Blood is a connective tissue with an extracellular matrix that is referred to as blood plasma or simply plasma. We find different types of cells and also cell fragments in blood. So let's clarify what it means. All these cells and cell fragments form inside the red bone marrow, which we know red bone marrow is found in the spongy bone. We have stem cells inside the red bone marrow that they can go through different processes and produce, for example, red blood cells, or we can say erythrocyte, white blood cells, or we say leukocytes, and also platelets, or I can say thrombocytes. We do not consider platelets cell. So when we say blood cells, we simply mean erythrocyte and leukocyte, red blood cell, white blood cell. Why we do not consider platelets cell? Simply because in the red bone marrow, at some point from those stem cells, we end up having some huge cells that we call the megakaryocytes. And at some point, the megakaryocyte explode into about 2,000 to 3,000 small fragments. And we name each one of those fragments a platelet. That's one of the main reasons that platelet is not a cell. It is a cell fragment. As you know, blood is denser and more viscous than water. It feels slightly sticky. The temperature of blood is 38 degrees of Celsius. That equals 100.4 degrees of Fahrenheit. So this temperature is about 1 degree of Celsius higher than oral or rectal body temperature. The pH of blood ranges between 7.35 to 7.45. So blood pH is average 7.4. And the color of blood, even though it's red, varies depending on how much oxygen we have in blood. When blood flows into pulmonary capillaries, which we have inside our lungs, oxygen diffuses into the blood and blood becomes saturated with oxygen. That's the moment blood has a bright red color. But when blood flows into systemic capillaries that we have near our tissues and oxygen from blood diffuses to the tissues, carbon dioxide from tissues diffuses into blood, basically blood is unsaturated with oxygen. That's the moment that the color of blood is dark red. And please note, in the procedures that we have in the medical field, we usually deal with the dark red, the deoxygenated blood. For example, when we have an intravenous injection, we inject the drug molecule into those blood vessels that carry deoxygenated blood, the dark red blood. Or when we want to get a blood sample, or when you're donating blood, again, the target would be the dark red, deoxygenated blood. Depending on male or female, the volume of blood is different. In an average size male, we have about 5 to 6 liters of blood. That equals 1.5 gallons of blood. In an average size female, we have 4.5 liters of blood. That equals 1.2 gallons of blood. When we analyze the component of blood, we notice that 55% of blood is the plasma. And please keep in mind, most of the plasma is water. To be more specific, 91.5% of plasma is water, 8.5% of plasma is solute. Most of the solutes that we find in blood plasma are proteins. And it is interesting that most of these plasma proteins are built by liver cells, hepatocytes. So that's something to consider in patients that they have any type of liver disease. One of the places that we see abnormality would be in the plasma proteins that we have in blood. When we focus on the plasma proteins in blood, we can categorize them into 3 classes. 54% of plasma proteins are albumins, 38% of plasma proteins are globulins, and 7% would be fibrinogen. For example, fibrinogen is considered one of the clotting factors, so it's involved in blood clot formation, the coagulation process. Speaking of globulins, we also have release of a type of globulin that is considered gamma globulin from some cells that we refer to them as either plasmacytes or plasma cells. So during immune responses, plasma cells release gamma globulins. We know gamma globulins as antibodies. So it's not surprising that we find antibodies in the plasma of an individual. Besides these plasma proteins, we have other solutes found in plasma. For example, we find nutrients, we find electrolytes, we find enzymes, hormones, gases such as oxygen, carbon dioxide, and waste such as urea, ammonia, uric acid, creatinine, and bilirubin. Clearly, one of the main functions of blood would be transporting nutrient and oxygen to the tissue cells and collecting waste from tissue cells, sending this waste to those systems of our body that can excrete the waste out of our body. As I mentioned, 55% of blood is plasma. The other 45% would be cells and cell fragments. But realistically speaking, when we focus on the 45% of the blood, we see that most of it, and by most I mean 99% of it, is red blood cell. Leukocytes and thrombocytes form less than 1% of the blood. Speaking of erythrocytes or red blood cells, in an average size male, we find about 5.4 million red blood cells in every microliter of blood. In an average size female, we find about 4.8 million red blood cells in every microliter of blood. When we refer to the percentage of blood that is occupied by red blood cells, in short, we refer to this percentage as hematocrit. Please note, inside this cell, we do have a molecule named hemoglobin, and hemoglobin is the red pigment. So we can say that in general, the reason red blood cell is red is because it contains hemoglobin, and the reason blood is red is because it contains red blood cell. Red blood cell is a cell that has two concavities. So the biconcave shape actually adds to the surface area of this cell, which allows the cell to have more surface area to diffuse more oxygen and carbon dioxide. Inside this cell, we cannot find nucleus. Also, this cell lacks most organelles, including mitochondria. We know that mitochondria is responsible for aerobic cellular respiration. So when we say red blood cell does not have mitochondria, it simply means that this cell is not capable of aerobic cellular respiration, the process that requires oxygen, which that simply means that red blood cell does not have any use for the oxygen molecules that it transports. And inside each red blood cell, we find about 280 million molecules of hemoglobin. In general, 33% of the red blood cell's weight would be due to the hemoglobin molecules that it carries. One important job of the hemoglobin molecules that we find inside the red blood cells would be carrying oxygen and carbon dioxide. When we focus on how blood transports oxygen, we see that 98.5% of oxygen inside the red blood cell binds to the hemoglobin molecule. 1.5% of oxygen is in plasma as dissolved gas. When we follow the transport of carbon dioxide in blood, we notice that 23% of carbon dioxide binds to the hemoglobin molecules that we have inside the red blood cell. 7% of carbon dioxide is dissolved gas in plasma, and 70% of carbon dioxide travels in plasma, but not in the form of the gas. It travels in plasma in the form of a buffer named bicarbonate ion. In case you're wondering, how do we end up with bicarbonate ion from carbon dioxide? That's an interesting story. Inside the red blood cell, we do have some enzymes that they allow carbon dioxide and water to react with one another. So when we are inside the systemic capillaries, the capillaries that we have within our vascular tissues, and carbon dioxide from tissue diffuses into blood, most of the carbon dioxide goes inside the red blood cell. Recall, 7% stays in plasma as gas, 93% goes inside the red blood cell. Out of the 93%, 23% binds to hemoglobin molecules, and 70% react with water inside the red blood cell. The result of this reaction would be carbonic acid. And then carbonic acid dissociates into two ions, hydrogen ion and bicarbonate ion. Most of the bicarbonate ion leaves the red blood cell and goes inside the plasma. That's how we end up with bicarbonate ion from carbon dioxide. So that would be one of the jobs of hemoglobin, to carry these two important gases. One other interesting function that hemoglobin has is its involvement in slowing down bacterial growth. One thing that bacteria need to grow and multiply would be iron ion. Hemoglobin is a molecule that carries 4 iron ions and is not willing to share that with bacteria. So having enough of this iron binding protein inside our red blood cells can actually help our immune system to slow down bacterial growth. So we can say that hemoglobin is involved in immune responses. That's actually one of our internal defense mechanisms. Another thing that hemoglobin can do would be it's capable of binding to nitric oxide. Recall that the squamous cells that line our blood vessels, and we refer to these squamous cells as endothelial cells, release nitric oxide. Nitric oxide is a vasodilator. Hemoglobin can bind to the nitric oxide released from endothelial cells, and then under some circumstances, hemoglobin releases the nitric oxide. And nitric oxide causes the dilation of the blood vessel, which this actually increases oxygen delivery to the tissue that is sitting close to the place that hemoglobin molecules release the nitric oxide. So we can say that hemoglobin has also a very important role in regulating the blood flow and blood pressure in our tissues. After this short summary about red blood cells, then I focus on thrombocytes or platelet, these cell fragments. When we count the number of platelets in blood, we see in every microliter of blood we have only 150,000 to 400,000 platelets. These cell fragments have very short lifespan. They can live 5 to 9 days, and then they will be removed by macrophages that we have in spleen and liver. Please keep in mind, red blood cells have a much longer lifespan. In general, most resources consider the lifespan of a red blood cell about 120 days, and then those red blood cells that are misshaped or they're worn out, dead, dying, will be removed by the macrophages that we have in red bone marrow, liver, and spleen. So in general, platelets are living considerably much shorter than red blood cells. One important thing that platelets are involved in would be the process to stop hemorrhaging, to stop bleeding. Because inside the platelets, we do have some tiny sacs filled with some chemicals, including some of our clotting factors. So when we start bleeding, which is due to the damage to the wall of the blood vessel, platelets will be activated in different ways, which in a separate video, I explained a few different methods to activate platelets. And then the active platelets start secreting those substances out, and they help initiating and progressing the process of blood clot formation. However, that's not the only thing that platelets are involved in. Please note that platelets can also release histamine. Histamine is one of the chemical mediators of inflammation, and we know inflammation is one of our immune responses. So platelet, aside from its involvement in hemostasis to stop bleeding, is also involved in our internal defenses. And one other thing that is good to note that is released from platelets would be platelet growth factors. These are some proteins that they enhance tissue regeneration. This leads us to a treatment plan that is called platelet-rich plasma therapy. In general, in those patients that they are dealing with a chronic injury to their, for example, tendon, ligaments, skeletal muscles, skin, let's say due to physical trauma, explosion, burn, and such, or they have a problem in wound healing, let's say the size of the wound or the depth of the wound is more than what body is capable of healing it fast enough, or even in male baldness, what we can do, we can actually inject a concentrated amount of platelet into the body of these patients exactly near the spot that we would like to speed up and enhance tissue regeneration. How do we end up with this concentrated platelet? We extract some blood from the patient, and then through the process of centrifuging, we separate plasma from cells and cell fragments. And recall that about less than 1% of blood would be white blood cells and platelets. We separate the platelets, and then we have a concentrated form of platelets, and then we inject that exactly in the area that we would like to stimulate the tissue regeneration. This is a well-known treatment, and that explains how important would be the role of this tiny cell fragment. The next group would be the leukocytes, white blood cells. Clearly, the main job of the leukocyte would be their involvement in immune responses. They use different strategies, different immune responses. However, one thing that I can say all of them have in common would be having these small granules in their cytoplasm. Inside these granules, they store some chemicals that they use in immune responses that they're involved in. When we want to look at a blood sample using light microscope, obviously we stain this light, and what happens is that the granules of these leukocytes have different attraction to the dyes that we use to stain this light. One group, their granules get the stain, so when we use the microscope we can actually see the granules. We name this group granular leukocyte or granulocyte. The other group, their granules are not attracted to the stain, so they don't really get the stain. That's why when we look at these white blood cells, we have difficulty to see the granules. So I emphasize, they do have the granule, it's just that their granules is not easily visible. We name this group that we have difficulty to see the granules agranular leukocytes or agranulocyte. The three granular leukocytes are neutrophil, eosinophil, and basophil. So these three are the granular leukocytes. The agranular leukocytes are lymphocyte and monocyte. And I walk you through each one of these cells. I start with the first responder, the one that usually when, for example, a tissue is under attack by microorganisms, migrate to that tissue. Neutrophil is the one. When I look at the nucleus of neutrophil, I see that the nucleus has 2 to 5 lobes. For example, in this neutrophil, I can count 5 lobes. As you see, the lobes are connected together. In this one, I can see 3 lobes. And then I pay attention to the cytoplasm. The granules get the stain, but since they are not very much attracted to either acidic or basic dye, they get a very light, or I can say even pale pink color. The next cell is eosinophil. I see that the nucleus has 2 lobes, and then inside the cytoplasm, because the granules are very much attracted to the acidic dye, they get orange-red color. And as you see, the granules are not really covering the nucleus. We can easily see the 2 lobes of the nucleus. The next granular leukocyte is basophil. Usually this cell also has 2 lobes. However, one easy way to tell this is a basophil is by looking at the granules. As you see, the granules, because of the basic dye, have a dark purple color, and as you see, they're even covering the lobes of the nucleus. So those are the 3 granular leukocytes. And then I focus on the agranular leukocytes. I start with the lymphocyte. First thing that I notice is the round, massive nucleus that it has. When I look at the cytoplasm, I see a very thin layer of cytoplasm around the nucleus, which has a sky blue color. And as you notice, no matter how much we try, we really do not see the granules. We know it has granules, but we don't really see them easily. And please keep in mind that lymphocytes are classified into 3 groups: T lymphocytes, B lymphocytes, and natural killer cells, which clearly the name natural killer cell tells me that these cells are expert in killing. Actually, they're very good in mass killing. The next agranular leukocyte is monocyte. First, when I look at the shape of the nucleus, either we can describe it as kidney shape or horseshoe shape. And then when I look at the cytoplasm, I can see that the cytoplasm has a gray-blue color. And again, we have difficulty to see the granules even though they are there. And please keep in mind that's monocyte that after immigrating into a tissue differentiate into a larger cell that is called macrophage. And recall that we have two groups of macrophages. One group is fixed. It means that it stays in that tissue and does its job, which one of the main job of macrophage would be phagocytosis only in that spot. But we have some macrophages that we consider them wandering. It simply means that they can wander around from one tissue, they can go to another tissue, and again, they take care of their duties as a macrophage. Please keep in mind, as I mentioned throughout the video, each one of these cells, or when we talk about molecules, they do have many jobs. So the only thing that macrophage does is not phagocytosis. For example, macrophage, if it's infected by a virus, can release some chemicals named interferon, which actually helps in slowing down viral replication. Also, we learned that macrophages are one of the three groups of antigen-presenting cells. So in short, each one of these cells uses multiple strategies to participate in the immune response. But one general rule is that out of these 5 groups of leukocytes that we talk about, most of them, after they immigrate, when we say immigration, that simply means how a leukocyte leaves the blood vessel and then goes to the tissue close by. That's the process of immigration. Most of these leukocytes, after immigration and going to, for example, a tissue that is invaded by microbe, they get involved in the fight with the microbe and eventually they die. So they're like soldiers that when they go to war, they know that there is no return for them. The exception would be for lymphocytes. I hope you find this brief information about blood interesting and helpful.